The overall goal of this procedure is to perform large-scale RNA interference screens in C. elegans using a bacterial dsRNA feeding library to knock down gene expression. This is accomplished by first culturing bacterial clones from a dsRNA library under growth conditions that prevent plasmid loss. Begin by duplicating each primary library plate and then using the 96-well duplicate library plate to generate temporary stocks of bacteria on agar omniplates. Bacteria from these omniplates are grown overnight in liquid culture. And subsequently transferred to the surface of worm feeding plates. Each bacterial clone from the library carries a unique dsRNA encoding plasmid that targets one C. elegans gene for knockdown. The second step is to place synchronized worms onto the feeding plates. Next, the worms are grown for several days in a humidified chamber at 20 degrees Celsius. As the worms feed on the bacteria, the induced dsRNAs are released into the gut of the animal, where it then spreads to surrounding tissues to cause systemic knockdown effects. The final step is to examine the fed animals.